Hi, my name is Lily Wang, and I'm a student at Ithaca High School. This summer, I was fortunate to be in the Hua Lab working under my mentor, Pei Tiaxie. My project this summer was dissecting calcium homeostasis and its roles in temperature response and immune signaling, and we did this in the model plant Arabidopsis. So some critical background to know is that in order for plants to be able to survive and thrive in their environment, they have to be able to sense what's going on externally and then respond and adapt to their environment accordingly. And plants are able to do this by sending cellular signals both within one cell and across multiple different cells. And these signals are passed along through the roles of different proteins and other small molecules. And we particularly focused on calcium because calcium ions are really crucial as a signaling molecule. So the first thing that we wanted to look at is how, well, the overarching goal is we wanted to look at how altered levels of cytosolic calcium in plant cells will impact their response to their environment. So the first thing we wanted to do is see how a lower level of cytosolic calcium will impact the temperature response of plants. So we created this lower level of cytosolic calcium by overexpressing a gene, CAX3, that encodes for this protein here on the vacuole that typically pumps calcium into the vacuole. And then we subjected these CAX3 overexpression lines to heat treatments and cold treatments. And we found that under heat treatments, these plants have increased survival rates compared to wild type plants, and that in cold treatments, these plants have a much smaller size compared to wild type plants. And then secondly, we wanted to look at how altered levels of cytosolic calcium will interact with their immune pathways. So first, we created plants that had an elevated level of cytosolic calcium, which we did by knocking out two transporters, ACA8 and ACA10, which are located on the plasma membrane, and they export calcium from the cell. So these double mutants, ACA8, ACA10, have been shown in previous studies to exhibit a much smaller size compared to the wild type plants. So we wanted to see how these plants will interact, how the altered calcium levels will interact with their immune response. So we created two triple mutants, one where we knocked out a gene called CBP60G, and we found that these triple mutants were much smaller in size. We also knocked out a different gene, a different immune response gene called SID2 in the same double mutant background, and we found that these plants were much larger in size and more comparable to the wild type plants. So overall, we found from this project that there's really complex interactions between cytosolic calcium and immune response, and further testing will happen to show the underlying mechanisms for what's happening. Yeah.
So what was one of your favorite discoveries in this project?
One of my favorite discoveries because, from reading a lot of past papers, I had seen that altered levels of calcium in plants typically cause them to die really easily when they're under temperature stress. So I had assumed that when we had lowered the calcium, that CAX3 plants would die a lot faster, but they actually look a lot healthier if you look in the picture. And that was really surprising, and that made me really happy to see the plants all nice and green.
Thank you so much.
Thank you.